Hello, everyone. Thank you for joining Tika Science News. We have the great privilege of being joined by two really remarkable doctors today, Dr. Eckenstead and Dr. Lewis, who recently published a paper about polyneuropathy in Siberian cats. And we're going to ask them, because they have all the definitive answers, the finite truth, the final answer to why any cat maybe walks funny. No, that's not likely, because there are no final answers. Just like anybody who breeds a cat, I'm breeding a cat for the cat that's in the future. If it were perfect, I would spay and neuter all my cats and never have to breed one again. But because this is a process and I'm working on going toward a goal that is way out there, they are part of this incredible scientific process of investigating, gathering information, and asking questions. And they've got some really good answers so far. So welcome, Dr. Eckenstead and Dr. Lewis. Thank you for joining us.
Thanks.
Thanks for having us.
So you guys published a paper about polyneuropathy in Siberian cats. Tell me why you decided to do that.
Sure. It actually is kind of an interesting story. It was a little bit fortuitous. So I'm on faculty here as a neurologist at Purdue, and we saw with one of my residents, who's actually the first author on the paper that you're showing there, Dr. Crawford, we saw a Siberian kitten that appeared to have neuromuscular disease, was weak in all four of his legs. And as we were getting started with doing just a diagnostic workup in that kitten, we realized, again, somewhat fortuitously from a conversation with another neurologist who practices in a different state, that she too had seen a Siberian kitten with similar signs. And so we then kind of put two and two together and did a little bit of legwork and realized that they were actually related cats. So that was kind of the jumping off point for us for why we wanted to look into it. And then we said, gosh, we're lucky here because we have Dr. Eckenstead, who's our geneticist. So reached out to her to say, hey, is this something that you think could be genetic in nature and that you'd be interested in chasing down with us?
Wow, and Chase, you did. So tell me about that process. Once you found these, or two kittens were identified with similar clinical symptoms, what do you do next?
Sure. So like I said, I was in charge of the clinical side, but Dr. Eckenstead can tell you kind of how things work from her perspective once she identifies a potential unique case or cases.
Mm-hmm. Sure. Yeah. So thanks, Dr. Lewis. We basically just start by contacting the breeder as much as possible, sometimes through Facebook. I mean, I can't tell you how well Facebook has assisted dog and cat genetic researchers. It's one of the probably most legitimate uses of Facebook for science that I can think of. It's a great way to connect with people. So we reached out to the breeder. And I should take a little aside here to say that every dog and cat genetic researcher that I know, when they work with breeders, we absolutely positively respect confidentiality. We never reveal names. We never reveal registered names. We never reveal kennel or cattery names. And that's partially because if we did, nobody would want to work with us. We understand that breeders are always concerned about the reputations of their breeding programs. And so we're always respecting confidentiality. And if you look at that paper, we did not reveal anything that should be identifiable. So we reached out to the breeder and ultimately were informed that there were not two, but four kittens from this related family that had shown very similar a similar clinical syndrome. And so what we do then is we start trying to collect DNA samples and also to put together that pedigree. So I don't know, Anthony, maybe you could show the pedigree from the paper?
Absolutely.
So we start putting that pedigree together. We look at the relationships between the cats that are affected and cats that are unaffected. We talk to each owner of each cat to find out, if that cat was normal or not normal. And certainly Dr. Crawford and Dr. Lewis did a lot of follow-up work in terms of speaking with owners to verify that each cat's health status. These sorts of things are really huge team efforts. Dr. Drager, who's also on this paper, was in my lab, and she used a neat program to build this pedigree. And what this shows here is the dark purple are 4 affected kitties. And then their parents are highlighted in the light purple. And we do that just because if this was truly a single gene thing, so like a brand new mutation that had just shown up and was being inherited through the family, those parents should be carriers or heterozygous. They're all healthy, but they have the ability to pass on this altered version of whatever gene it is that's creating this clinical syndrome. And so we put the pedigree together and we look for a couple things. We look for ratios, how many affected versus unaffected kittens are there in any given litter. We look for inheritance patterns. You know, are there a bunch of cats in multiple generations all affected, or does it kind of show up more surprisingly from unaffected parents, which is what we're seeing here. And that's kind of the classic recessive condition look. And then we look to see if maybe there's a way we could trace that potential mutated or altered version of the gene back. So that yellow cat is, and we can never prove this because we don't have the DNA from that cat. I don't even think that cat is still alive. That maybe that was where the brand new mutation may have shown up. You may have seen similar sorts of things when you look at hemophilia in the royal families of Europe, and people have tried to trace that back and figure out where did that first mutation come from. So what we know so far is that this pedigree looks like a recessive condition, meaning that it looks like it's just one gene and it looks like it's new. And I should take another little aside here and just say, you know, these sorts of things happen, right? They happen by chance. Our genomes are rearranging themselves every time we reproduce, right? So sperm and eggs are never going to be exactly like the original animal they came from, and that produces variety. But our cellular machinery can make mistakes. And so these sorts of little genetic changes that end up creating disease can happen. Absolutely. And it's not, what I like to say is it's not your fault, right? Like these things happen randomly. And actually the breeders here have done a great job because we're working with them and we're going to try to eventually solve this. And then, you know, if we can get rid of it, right? Rather than trying to ignore it or hoping that it will just go away. So I want to point out that these things can happen to anyone, and that nobody should be blamed. And that when you can, it's best to try to work with a genetic researcher to try to solve them. I'll quit jabbering now.
No, I think it's really important that that all the points that you presented are really valid. And like when a new color is identified or a new pattern, or no one can necessarily take credit for creating that new color, because it's a combination of genetic anomalies, mutations, abbreviations, deletions, substitutions, switches that occur that we might appreciate, we might want to make more of it, but thankfully we can't take either credit or blame for it. And that's right. An aspect of the work that you guys do.
Yeah.
So how did these cats present to you, Dr. Lewis, when you saw them? Did they walk a certain way? Did they fall frequently? How did they look different?
Yeah.
And clarify the clinical presentation.
Yeah, absolutely. So the short answer is that it's a little bit variable cat to cat, but they all had some degree of weakness. So whenever we're doing our neurologic exam, so if a case presents to me for possible neurologic disease, the first question I always ask myself is, does it have a neurologic problem? Because you can have trouble walking if you are lame from a pulled muscle or something like that, right? So first question was, do these animals look neurologic? The answer to that was yes. But then the next part of our neurologic exam is to try to say where within the nervous system is affected. And the majority of the cases that we see, both dogs and cats, they have their central nervous systems affected, so their brain and their spinal cord. But uncommonly, we see animals with neuromuscular disease, and that's what these cats fell into that category, which is always a little bit exciting because we just don't see them as frequently. And then within the neuromuscular system, our clinical exam doesn't necessarily help us beyond that, but we can try to say, do we think it's the muscles affected, the nerves affected, or where the junction, where the nerves and the muscles come together and talk to each other. But all of the cats had a degree of weakness with preserved proprioception, or what we call like knowing where their feet are in space. If you've ever seen a neurologic exam performed and they turn the animal's toes over, a normal animal should recognize that that's abnormal and turn it over quite quickly. And with most of our neuromuscular diseases, they can be very weak, but that proprioception is preserved. And that was the case in all of these cats. And most of them had just their pelvic limbs that were weak, and their thoracic limbs were relatively normal. One cat, all 4 limbs were quite weak, but was worse in the pelvic limbs. And one of the most common presenting complaints for the owners was difficulty jumping. So these were young cats. And so they're, had been very active, and they would jump on and off the counters and the kitchen, the furniture and things like that. And so they were having difficulty where they were, especially because their pelvic limbs were so much weaker, and that's the launching off point for most cats. And so one of the first things that their owners noticed was difficulty jumping, and that they weren't walking normally. So they were both kind of only walking short distances before they would seem tired and sit down. And their legs, they don't walk on the bottoms of their feet like we do. They walk kind of up on their fingers as a normal cat. And so their ankles were dropped down as well, what we call a plantigrade stance. And that's quite abnormal for a little kitten. So that's something else that they noticed and that we could see as well.
Wow, well, that is super. And I have to confess, I raised Bengal cats and have for 28 years or so. And polyneuropathy exists in Bengal cats. And I've for a long time thought, well, it's inherited, and we just don't have all the resources to investigate it properly, and send sample to Leslie. And but now there are people like yourself out there who are really going for it. And often it was misdiagnosed. So sometimes they were diagnosed with FIP, sometimes it was just generalized ataxia. How can an owner or a veterinarian specifically diagnose polyneuropathy from other conditions?
Yeah, so it actually takes a fair bit of work. Like I said, the majority of neurologic cats and dogs that we see have a problem with their central nervous system. So most veterinarians aren't used to seeing neuromuscular cases, of which polyneuropathy would be a subset underneath that. And so it's quite a bit, it's easy to miss, I think, because people aren't as familiar with it. And it's not something, because it's uncommon, that we spend a lot of time teaching our vet students about. So those out in clinical practice, understandably, don't have as much experience. So that's where we come in as neurologists. We always love to kind of hear on the phone, do consultations over the phone, watch videos, or ideally get those cats to come into a clinic where there is a neurologist that can examine them. But our exam can only take us so far. So then next steps for diagnosing animals with neuromuscular disease are usually electrodiagnostics. Those are functional tests that allow us to assess how those nerves and muscles are working. And that's what, the figure there shows us evaluating how we evaluate the muscles, and then the lower panel is looking at the function of the nerves, how well they're conducting messages along the nerves. And that gives us kind of confirmation that we're in the right area, that it is in fact the neuromuscular system that's affected. But it's not usually very specific. for most neuromuscular diseases, we can get some kind of indication that it maybe is more likely this than that. But if the nerve is abnormal, it's going to react the same way, whether it's, let's say, an inherited process or an infection or something of that nature. So then we typically take samples of a couple different muscles and a small piece of nerve. And that's safe for us to do without actually affecting the function. So we can kind of take a small little sliver of the nerve and some pieces of muscle, and we usually send them off to our collaborator, Dr. Shelton, at the University of California, San Diego. She is kind of pretty renowned in our field for her neuromuscular lab, and so did the evaluation of those samples for us. So you can see there in the figure. And that is additionally helpful for us to kind of further dial down and complement that electrodiagnostic data that we got. And sometimes it gives us a definitive answer. Other times it helps us rule some things out. But then we're still left with a little bit of more work to do to try and say, okay, well, we've got these changes that might be secondary changes. Let's go try to look for why they're occurring.
Yeah. So where do you go from here in your research?
Yeah, I'll let Carrie take that one.
Sure. So yeah, we wanted to get the paper kind of describing the clinical condition published first, and so we've done that. And really the next goals are to try to solve the genetics of this condition in the Siberian forest cat. So we picked out, all 4 of our affected cats, and we did get DNA from all of them. And then we picked out 4 related cats that were normal. And we have generated whole genome sequence. So that means we sequence the entire genome, which is a lot of data. Remember, the genome is 2.5 billion letters long. And so we covered that about 30 to 40 times for each cat. So all of the analysis of this data has to happen on campus supercomputers. I have a graduate student who's working on that now. So it's a lot of data, and it takes a while to even get it kind of ready to look at. We have to trim it, we have to align it to the reference cat genome so it gets assembled correctly. And then the goal is to start looking for any variants or changes, and you mentioned several of the different types, Anthony, the deletions, insertions, single letter changes, those sorts of things, and then see if we can find a variant like that is, if it was really recessive, which is what the pedigree suggests, that would be homozygous in all 4 of the cases and heterozygous in all, so carrier, 1 copy instead of 2 in all of the parents, and potentially in some of the litter siblings that were normal, or else homozygous for the reference or unaffected normal letter or condition of that area in the genome, basically 2 copies of normal. So that's the search we're currently in. We're working our way through that. We would eventually, if we were able to identify a specific variant in a specific gene, assuming that it made sense. And I have worked in neurogenetics long enough to tell you that there's a neurological gene almost everywhere you look in the genome. Honestly, that's the truth. Our nervous system takes a lot of good fine-tuning by our genes. But if we were able to identify something that looked like it was associated with the condition, then the next step would be to test all the rest of the cats that we have DNA from in that family, because we have DNA from more than 8 of those cats. And then we would move further and start to test the rest of the breed. So we would want to know how common is this variant or mutation in the breed at large. We would probably also screen maybe 100 or so just typical cats. We can access leftover blood samples right here at the hospital, where we could do something like that and just test, random bred cats, or 1 or 2 of the, occasional purebred cat that actually does come through the clinic. I think we have a Siamese, we have an Osa cat, we have a few other, Bengals, unique breeds, Tonkinese, but mostly random bred cats for that. And so we would just screen and make sure that this isn't a mutation that's out there in cats, like basically just ways to make sure it was real.
Mhm.
And then assuming all of that held up and we were pretty confident that it was the right thing, then we would look into, how can we offer this as, a tool for breeders to use in matings in order to, avoid producing more affected cats. It's not something I would expect to necessarily be applicable to Bengals. I know Bengals have, as you mentioned, Anthony, that they've certainly been well documented as potentially having a genetic form of a polyneuropathy. But if we found something and could solve this genetically in the Siberian forest cats, my predictive guess is that it probably will be private to that breed. And so even if we could develop a genetic test that would help Siberian forest cat breeders, it's unlikely to be applicable to other breeds like Bengals.
Well, that's a very good point. And the work that you guys are doing is quite amazing. The quest, both the identification and then the search for if it exists throughout the random bred cat population in Siberians, and then if even related breeds from that area of the world and the rest of the cat population is really fascinating. So what can breeders, cat owners do if they want to assist you guys? Can they send you DNA samples if they have a Siberian that walks fine or walks funny or has been diagnosed or with polyneuropathy or is related to any of those cats? I can tell, before you answer, that the International Cat Association, we're really excited about empowering and encouraging folks to participate in clinical research and genetic research with the academic community. And so anybody who does decide to participate in your research in the future, they'll get what would equate to a gold star and some special rewards and benefits from the International Cat Association for just participating, because that's how we find the answers. And
Cool.
we give you guys new questions too, like Dr. Lewis mentioned. New questions is important.
Yes. So absolutely, we would love to get. We would be absolutely willing to take samples from additional Siberian forest cats, whether they were completely normal or, and we would be very keenly interested in hearing if there are any other cases, affected cats out there. And then we would work with that owner and that breeder, of course, privately and confidentially to see if they fit into the same pedigree, which also helps us figure out if this is for sure the same thing or if it's maybe even something different. It's scary to suggest it, but it's not unreasonable for multiple genetic neurological diseases to show up even in a pretty tiny breed. The example, I'll switch to dogs, the dark side of the forest here. I've worked quite a bit with a large team of international amazing researchers on the Leonberger dog breed, which is a numbers-wise pretty small breed, body-wise large. But we've solved 4 neurological conditions, genetic neurological conditions in that breed so far. And so the point is that we don't know, right? And as Dr. Lewis said, it could be that even if we did solve this particular genetic thing in the Siberian forest cats, it may not answer every case, even within that breed. So yeah, we would love to take samples from normal Siberian forest cats, affected Siberian forest cats, and then continue on, the journey of trying to solve this, or potentially if anything new came up. I know there are some other fantastic cat genetic researchers out there, and try to be, we play nice. So I'm not going to step on anyone's toes trying to take over other projects, but when new ones come along and if we have the opportunity to work on them, we're really happy to do that. And that's wonderful that TICA has a way to reward people for participating. I love that.
And the benefits affect us all, and we all want happier, healthier, more beautiful cats. And because my version of beauty may not necessarily be what Siberian owner's version of beauty is, why don't we just encourage everybody to keep going in their direction? And your help, we can get there sooner, faster.
Yeah. I'll say too that even on the clinical side, that we're always happy to see a cat, a Siberian kitten that is showing similar clinical signs here. But we have a nice network of neurologists, veterinary neurologists out there around the country, around the world. We maintain like a very active listserv ourselves. So other neurologists can always contact us as well, or the owners can contact, somebody that they don't have to come to our hospital necessarily to be seen, to still provide meaningful information to help us with the project. So
that is awesome. I don't want to take up all of your time today, but I'll tell you this issue of our science newsletter includes your study and also Emily Groff from Emory, who identified PEA15 deletion that caused a cerebral dysgenesis, or juvenile cerebral dysgenesis. So the kittens kind of shook and they had tremors as kittens. They kind of grew out of it, and then it came back when they were past 5 years old, and part of their cat colony population at Emory. Yeah, so we talk about a small population, random bred cats that create their cat population. So it's so important that we collaborate with you guys, and it's fantastic you're collaborating together.
Yeah.
We appreciate you. Any last words for the cat lovers out there, Dr. Lewis?
Okay. Well, I was going to say, I wore my pin. You can't really see it, but it says, "The future is feline."
Hey, I agree with that wholeheartedly. So you've worked with some big dogs, but what do you have to say to the cat owners out there?
Oh my goodness. Well, I kind of laugh about that because I've done more of my work has been in genetics of dogs than cats, although we're increasingly working more on cats here as well. But actually, I don't own a dog. I have 2 cats. And I've always been I think the cat and I have a much more synchronous lifestyle. And if I could be my cat and just sleep all day, I think I would sign up for that.
I am down with that, but I am so glad that you don't, and that both of you continue to work with folks like myself and your colleagues around the world to find and fund. That was my phone. It's the Washington Post about the next interview we're going to do. But thank you guys so much. Have a terrific day, and thank you from behalf of cat lovers everywhere.
Yeah, thanks for having us.
Thanks for having us.
All right, I'm going to stop recording now, but.
And certainly if anyone has questions, I can't make any medical recommendations for a cat I haven't seen, but certainly I can talk to their referring vet and things like that. So certainly welcome to provide my email address as well.
Yeah, and if you are a Siberian cat owner, or you're a Siberian cat adjacent and can influence that cat's owner in any way and want to participate in the genetic research, submitting us the sample is pretty easy. So just email me and I can give you all the information, all the forms that have to be signed and that sort of thing. My email address is kje0003@purdue.edu. That's Purdue, Purdue. So KJE0003@purdue.edu. And I can give you more information.
Super. And it's also going to be on tika.org. If you click the science newsletter, Dr. Eckenstedt's email and Dr. Lewis's email will be in the bottom of that article. Thank you both so much. Have a terrific, terrific afternoon.
Thanks.
Thanks.